Hi, in this video we're going to be looking at mitosis, which is a type of cell multiplication. Cells multiply themselves for many different reasons, such as growth, repair, cloning, and asexual reproduction. There are two different ways that cells can divide themselves. These are mitosis and meiosis. We're going to be looking at mitosis in this video, and there's going to be another video for meiosis. A good example for mitosis is the copying of a fertilized egg. A fertilized egg is known as a zygote. Let's have a look at human cells in more detail. Humans have 23 different chromosomes. Gametes contain one of each of the 23 chromosomes. They're known as haploid cells, and examples are eggs and sperm. A fertilized egg is a diploid cell because it has two sets of each of the 23 chromosomes. It has one set from the sperm and another set from the egg. After an egg has been fertilized, it will divide into 2 cells, then 4, 8, 16, etc., etc., and this process will continue until the adult body contains millions of cells. Mitosis is the process that's responsible for the fertilized egg dividing into 2 cells, then 4, then 8, then 16. When a cell divides by mitosis, 2 cells with the same number and type of chromosomes of the original cell are formed. The mitosis process is responsible for multiplication of all of our cells in our body except sex cells. Sex cells are known as gametes, and they are eggs and sperm. So let's have an overview of what mitosis is. Here we have a cell, and this cell is going to be divided into 2 genetically identical cells. The cell that is dividing, which is the original cell, is known as the parent cell, and the 2 cells that are produced are known as the daughter cells. Both daughter cells are genetically identical to the parent cell, and to ensure that this is the case, the daughter cell must do 2 things. Firstly, the parent cell must copy each chromosome before it divides, and it must divide in such a way so that both of the daughter cells contain 1 copy of every chromosome. If the parent cell did not copy all of the chromosomes, or it didn't split correctly, it would mean that the daughter cells would not be identical to the parent cell, and they won't contain all of the necessary genes. So that's like a general overview of what mitosis is. Now let's have a look at mitosis in more detail. There are 4 different stages of mitosis, and we're going to be looking at the different stages of mitosis on this parent cell here. This cell has 4 chromosomes. The first stage in mitosis is the prophase. During the prophase, the cell copies all of its genetic information. This is so that the genetic information is the same in both the daughter cells and the parent cells. When a cell is not dividing, the DNA in the nucleus will be in long strands. However, when a cell is dividing, the DNA will coil up, forming chromosomes. The DNA in our cell that's in the example has already coiled up to form chromosomes. The chromosomes will then copy themselves. The original uncopied chromosomes are known as chromatids. Chromatids are 1/2 of 2 identical copies of a replicated chromosome. The chromatids are joined together by a centromere. So if we look at the diagram, if we look at the green chromosome, we can see the 2 chromatids and the centromere, which is the white circle in the center. At the end of this stage, the membrane of the nucleus breaks down. The next stage in mitosis is the metaphase. During this stage, a structure called the spindle forms. The chromatids are attached to the spindle by the centromere, and the centromeres start to release spindle fibers that go either side of the spindle. So on the diagram, we can see the spindle. We can see that the chromatids are joined to the spindle by the centromere. We can also see that the centromere is releasing spindle fibers to opposite ends of the cell. This brings us on to the 3rd stage of mitosis, which is the anaphase. During this phase, the spindle fibers shorten, which pull the chromatids of each chromosome to opposite ends of the cell. Opposite ends of the cell are also known as the poles of the cell. So we can say that the spindle fibers shorten to pull 1 chromatid to each pole of the cell. The final stage of mitosis is known as the telophase. It is where the cell membrane and cytoplasm divide to form 2 identical cells. These 2 cells are known as daughter cells. The cells are identical because the spindle fibers have pulled 1 chromatid to opposite ends of the cell, and when the cell divides through the middle, there is one copy of each chromatid in each of the daughter cells. So let's have a quick overview of the 4 stages of mitosis. So here we have the parent cell, and this parent cell is going to divide into 2 daughter cells. The first stage of mitosis is the prophase. During the prophase, the cell copies all of its genetic information and the membrane of the nucleus breaks down. The next stage is the metaphase. During this phase, the chromatids line up along the spindle and they release spindle fibers to opposite ends of the cell. The next stage is the anaphase, and this is where the spindle fibers shorten, which pull the chromatids of each chromosome to opposite poles of the cell. The final phase is the telophase, and this is where the cell membrane and cytoplasm divide to form 2 identical cells, and these cells are known as daughter cells. One way to remember the phases in order is to use a PMAT. P for prophase, M for metaphase, A for anaphase, and finally T for telophase. So let's have a quick recap as to what the 4 stages are. The first stage of mitosis is the prophase, and during this phase the cell copies all of the genetic information. The next stage is the metaphase, and during this stage the chromatids line up along the spindle. The centromere, which is what the chromatids are joined together by, releases spindle fibers, and these fibers go to opposite poles of the cell. The third phase of mitosis is the anaphase. This is where the spindle fibers shorten, which pull the chromatids of each chromosome to opposite poles of the cell. The final stage of mitosis is the telophase, and this is where the cell membrane and the cytoplasm divide to form 2 identical cells, and these identical cells are known as the daughter cells. The final section of this video looks at the frequency of mitosis. The frequency of mitosis depends on where the cells that we are concerned with are located. For example, when we touch something, we lose thousands of cells that need to be replaced. The replacement is conducted by cells that are located underneath the surface, constantly dividing. Cells in the spleen and cells in the gut also have a high rate of mitosis. This is because cells in the gut are scraped off by passing food, and cells in the spleen are worn out by passing red blood cells. This brings us to the end of this video. For more content, quizzes and past papers, go on to www.elevise.co.uk.